In this video, we're going to discuss a topic that is all around us, but invisible to the naked eye. Let's rewind to the 1800s. Scientists were discovering that tiny organisms like bacteria caused a certain set of diseases. But there was something strange in the equation. They couldn't figure out what was behind illnesses like tobacco mosaic disease, which is a disease in plants. Even with a microscope, they just couldn't see any bacteria causing it. Then, in 1892, there was a scientist named Dmitry Ivanovsky. What he did was he filtered sap from infected tobacco plants. Sap is actually the fluid that circulates within plants, carrying water, sugars, and nutrients throughout it. It's similar to blood in animals. So when he filtered that sap, removing bacteria-sized particles, somehow the disease was still there. I mean, this was a bit strange because at that time the smallest particles known to cause diseases were bacteria. And now you're telling me that bacteria got trapped in the filter, but some particles that are even smaller than them were able to escape and remain there in the sap. This strange finding led to the idea of a whole new type of pathogen. It wasn't until the invention of electron microscopes in the 1930s that scientists could finally see these incredibly tiny organisms, viruses. Now, viruses come in a wide range of sizes. The smallest ones, like the parvovirus, can be as tiny as 20 nanometers, while larger viruses, like the pox virus, reach around 250 nanometers. To give you an idea of how small that is, viruses are typically 10 to 100 times smaller than the most bacteria. And because they're so tiny, they can pass through filters that would normally catch bacteria, and we now know that this was how scientists first realized that viruses existed, when they noticed diseases spreading through filters which were designed to stop bacteria. And here's another unique trait of viruses. They cannot grow on artificial media. You can grow bacteria in a petri dish with the right nutrients, but viruses? No chance. They need a living cell to survive and multiply. That is the reason they're called obligate intracellular parasites, because they need to enter a living cell to survive and reproduce. Unlike bacteria or other organisms that can live and multiply on their own, viruses cannot do anything whatsoever outside a host cell. They just don't have the machinery to make energy or build new virus particles on their own. Instead, once inside a cell, they hijack its resources, forcing the cell to produce more viruses. So this parasitic relationship makes them completely dependent on a host, which is why they're referred to as obligate intracellular parasites. On top of that, viruses are resistant to many common antibiotics like penicillin and streptomycin. These antibiotics work by targeting bacteria and destroying them, but viruses are not scared of them. They don't have the cellular parts, which means that the standard antibiotics are useless against viral infections. That is why treating viral infections often requires a totally different approach, like antiviral drugs or vaccines. Now let's move on to the shapes of viruses. They come in a variety of shapes and structures. For example, animal and plant viruses can be helical with a spiral shape, polyhedral, meaning they have many sides, spherical, a sphere-like shape, and complex, with a combination of shapes. Each shape serves a purpose. For instance, the tobacco mosaic virus, which infects plants, is rod-shaped. That is, it falls in the category of helical. Adenovirus is polyhedral. The poliovirus, which affects humans, is spherical, while bacteriophages, viruses that infect bacteria, have a distinctive tadpole-like appearance, which means that they have got a complex structure. The complete, mature, and infectious particle is called a virion. It has two main parts, the core and the coat. The core is the inner part of the virion and holds the virus's genome and various proteins that are important for causing infection. Genome is the virus's genetic material, which can either be DNA or RNA. It can be single-stranded or double-stranded depending on the type of virus. This genetic blueprint is what the virus injects into a host cell to begin the infection. Then we've got the core proteins. These are one or more proteins or enzymes that help the virus function once inside a host. These enzymes can aid in the virus's mode of action, making it easier for the virus to take control and replicate within the host. The next part is the coat, which is the outer layer. The coat gives the virus its shape. It is made up of the capsid, which is the protective protein shell surrounding the core. It's composed of repeating protein subunits called capsomeres. Different viruses have a differing number of capsomeres. For example, the herpes virus has 162 capsomeres, while the adenovirus has 252. These capsomeres fit together like puzzle pieces to form a strong protective layer. Some viruses, like the flu virus, have an additional lipoprotein envelope around the capsid. This envelope also has glycoprotein spikes. Now, what are these glycoprotein spikes? Well, this is something that helps the virus recognize and attach to host cells, making it easier to infect them. Think of these spikes like keys that fit specific locks on host cells, allowing the virus to enter. Let's now talk about the classification of viruses. These can be grouped based on the type of genetic material they carry, meaning that either they have DNA or RNA, and that material is either single-stranded or double-stranded. Another way to classify them is by their structure. Some viruses have an outer layer or envelope, while others are unenveloped. One group is retroviruses, which are RNA viruses with an envelope. These viruses use an enzyme called reverse transcriptase to copy their single-stranded RNA into double-stranded DNA. But why are they doing this? What's the need of it? Well, this conversion allows them to insert their genetic material into the DNA of the host cell, meaning that now they can become part of the cell's own genetic code. Examples of retroviruses include HIV, which causes AIDS, and hepatitis B, which infects the liver. Another type is the reovirus. Unlike retroviruses, reoviruses have double-stranded RNA and are shaped like an icosahedron, a 20-sided structure that provides stability. Reoviruses can infect plants and animals. In humans, these viruses can cause illnesses like Colorado tick fever. Viruses with small genomes, like the parvovirus, carry only a limited amount of genetic material. Parvovirus is a small DNA virus, which can be single or double-stranded with an icosahedral shape and a diameter of only about 20 nanometers. It can cause diseases like slabbed cheek syndrome, which often starts with a red rash on the face, along with symptoms like fever, runny nose, and headache. In adults, the rash is less common and joint pain is more likely to occur. Then we have some viruses that have medium to large genomes made of double-stranded DNA. A common example is herpes virus, a large DNA virus with an icosahedral shape. This virus group is responsible for causing a variety of illnesses in humans, including herpes, shingles, and even poxes. That's it for today. See you in the next one, and don't forget to subscribe. Thanks for watching.